Okay, so thank you everybody for this the second webinar of this new season of ILS webinars and the first webinar of 2024. I'm happy to be here with Marion Prudent, which is senior researcher at the Agroecology Institute in Dijon, France, part of INRA. Marion has been working a lot and focusing research on stress, on legumes, drought stress and abiotic stress, and also on the interaction between root system and soil microbial communities. And at this point, I would like to thank her and give her the virtual microphone, so she can talk today about peer resilience to drought. Thanks, Marion. You have the stage.
Okay, thank you, Nelson, for introducing me. So yes, I'm working in the agroecology department, and I am leading the grain legume ecophysiology group. And today I'm very honored to present the work of the group on the subject of peer resilience to water deficit. So this work comes against a backdrop in which agriculture is facing a major challenge that is feeding a growing population that will reach more than 9 billion people by 2050. So it means that we need to increase the production by more than 50% when compared with 2005. And against the backdrop of climate change that is characterized by rising temperatures, episodes of drought, or sometimes flooding, and also rising atmospheric CO2 levels. So legumes have a key role to play in this context, in an agroecological context, because they can provide a number of ecosystem services, such as the production service, provided by the protein-rich nature of these crops, and the support service provided by the symbiotic fixation of atmospheric dinitrogen, and the regulation service provided by the regulation of bioaggressors through the cycle cutting effect in rotation, and also the service provided by the storage of carbon in the soil, and particularly in permanent grasslands. And because of the various ecosystem services provided by the legumes, we can also say that these species have a major role to play in meeting the climate challenge. In fact, here we are, the supply of proteins means that we can switch to diets that consume less animal protein, while legume crops do not require nitrogen fertilizer and reduce the use of pesticides for the following crop. And all of these factors that can contribute to mitigating climate change by reducing greenhouse gas emissions. But the problem is that if we want to get these services, these ecosystem services, we must not only mitigate the climate change, but these species also need to adapt to it. So, for example, here in France, but it's also the case in other producing countries, still legumes yields are highly unstable. For example, you see here until year 2000, genetic progress enabled pea yields to increase, whereas since 2000, despite significant genetic progress, we have yields that have fallen by around this value, so it's negative. And you can see that during this period, we have different years with several abiotic stresses. So in you see that in red, it's when the year in a year when we had heat stress, in blue water stress, and in yellow when we have double stress. So we can see that these stressful events are occurring much more frequently in recent years. And in our department, our colleague Christophe Lecomte, maybe some of you know him, he has identified yield limiting factors in spring pea using a method known as drought. And he found that the main limiting factors were water deficit or excess water in the vegetative phase, also nitrogen deficiency between flower initiation and flowering, and high temperatures and water deficit during seed feeding. So this study revealed that there's a high need to improve the nitrogen nutrition of spring pea under fluctuating water condition, dew condition throughout the cycle. What about this nitrogen nutrition? You know, most of you know that this nitrogen nutrition is based on two different types of uptake. The first one is mineral nutrition and the fixation of atmospheric nitrogen within two underground organs. Mineral nutrition is taken up by the roots, while the nitrogen is fixed in the nodules in a mutual symbiosis with soil bacteria called rhizobia. And the idea is that the plant supplies energy to the bacteria, and in return, the bacteria supply ammonium to the plant. And these two modes, two types of nitrogen nutrition are complementary for the acquisition of nitrogen, but fixation is inhibited by the presence of nitrates in the soil. So here you can see that in the field, when the mineral nitrogen are very low, it's here, the plant set up nodulation, enabling symbiotic nitrogen fixation. And the greater the amount of the mineral nitrogen in the soil, the less the plant will use symbiotic fixation. This is explained by the fact that these two organs, that are roots and nodules, are in competition for carbon. In fact, symbiotic fixation is associated with a high carbon cost for the plant in order to allow the nodules to develop and to function. So here we can say that 4 to 6 grams of carbon are needed for the fixation of 1 gram of the nitrogen. This competition for carbon will lead to a modulation of root architecture. On the left, you have a root system of a pea plant that acquires nitrogen exclusively through the uptake of mineral nitrogen. And on the right, it's the root system of the same genotype under the same climate condition, but when the plant acquires nitrogen solely through symbiotic fixation. So clearly, you can see that the root architecture are very different according to the type of nitrogen nutrition. This is why we want to identify levers for action to enable legume resilience under water constraints. These levers could be genetic or agronomic. And to identify them, we need to understand the processes underlying the resilience. So this diagram shows a leguminous plant, a legume plant interacting with soil microorganisms, whether symbiotic like under symbiotic, I mean rhizobia or mycorrhizal fungi, or non-symbiotic. And when there is a water deficit, the plant changes the way it takes up and uses its resources, so water, carbon, nitrogen, or other mineral elements, and thus modifies the soil under the influence of the resources. Yeah, so here you can see where the water stress can impact everything. So in order to identify the processes underlying legume resilience, we wanted to understand first the importance of the nitrogen nutrition type, either by mineral nitrogen uptake or by denitrogen fixation, and also the importance of the plant phenological stage during which drought occurs. And then we wanted to assess the importance of stress intensity on these processes. Then we wanted to explore the genetic variability of plant responses to drought. And finally, at the end, we also wanted to explore the agronomic level, and to that aim, we concentrated on one facet, that is the microbial composition of the soil. So I will start with these two first points to give you an overview of the work that has been done to characterize the response to water deficit, and with first an overview of what happens depending on the nitrogen nutrition and the phenological stage of the plant. So to do this, a study was carried out on pea plants in interaction with a Rhizobium strain, in which the nitrogen nutrition mode was varied. So no nitrogen, mineral nitrogen, for plants whose nitrogen acquisition was based exclusively on denitrogen fixation, or with mineral nitrogen in excess. So the denitrogen fixation was inhibited and the nitrogen uptake was only based on mineral nitrogen uptake. So water stress was applied for 2 weeks at 3 different phenological stages. So the first one here was the flower initiation, or at flowering, or at the end of limiting stage of seed abortion. After each period of stress, the plants were fully rewatered, and then we explored the yield components at physiological maturity. So what we observed was that at physiological maturity, the water stress has a much greater impact under denitrogen fixation condition here, so with no nitrogen, mineral nitrogen, when it occurred at flower initiation, with a drop in yield of minus 40%, compared with only 20% when the water deficit occurred at flowering, it's here. And on the other hand, when nitrogen acquisition is based solely on the uptake of mineral nitrogen, the water deficit with a greater impact is that which occurs during seed filling, here with minus 20%. So this study enabled us to position the research question related to the water deficit during the vegetative period, here, and on the type of nitrogen nutrition based on denitrogen fixation. Yeah. So then we wanted to assess the response of pea plants when the intensity of the water deficit was varied. To do this, we subjected pea plants interacting with rhizobia to 7 levels of water availability, and we characterized the plant at the end of the water deficit period here, and after a period of rewatering. So at flowering initiation, 2 weeks of water deficit followed by 2 weeks of fully rewatering. So here we are able to show that at the end of the water deficit, the plants reduce the quantity of carbon fixed here, and that the allocation of this carbon between above ground and the root parts is modulated. In fact, carbon is preferentially allocated to the root compartment. And within the root compartment, we have 2 organs. We have the roots and the nodules. And concerning the roots, the growth and the number of the roots are not affected by the water deficit. And concerning the nodules, only the growth is affected and not the number of the nodules. So this leads to a decrease of the quantity of the fixed denitrogen, and with also a decrease on the specific activity of the rhizobia. Then, during the rewatering period following the drought, so it's on the right part of the diagram, the total, yes, what I wanted to say before is that we had exactly the same response, whatever the level of the water deficit, but what we observed was that the intensity of the response increased with the intensity of the water deficit. So the more intense was the water deficit, the more intense here, for example, was the decrease in shoot biomass. Okay, so then during the rewatering period, after 2 weeks of rewatering, the total quantity here of carbon fixed is still lower than in under the well water control, but the allocation of carbon within the plant returns to normal. So here the ratio, yeah, returns to normal. And after that, concerning the roots, you remember that the growth was maintained during the drought period. Here, during rewatering, we see that their growth and development is reduced through a decrease of the growth, but also on the number of the roots. And this allowed benefit actually of the nodules, because after rewatering, we see that the growth of the nodule is maintained and the number is not changed anyway. So what we can see also is that the fixation activity returned to levels that are similar to the well water control, and it results in finding a fixation that fully recovered after 2 weeks rewatering. And in the same way as during the water deficit, we've seen that the intensity of the effects was observed to increase with the intensity of the water deficit in the same way as during the drought period. And what we were able to observe is that we found that the establishment of new nodules during the rewatering period depended on the plant's nitrogen status at the end of the water deficit. So it means that the plant is able to adjust the number of nodules to the nitrogen needs of the plant. So we found this relationship for all the intensity except for this point that correspond to the highest water deficit intensity. When the water deficit is too intense, there's no relation anymore in adjusting the nitrogen, the nodulation to the nitrogen status of the plant. So then, in order to assess whether these responses were generic, we characterized the genetic variability of resistance to water deficit. To do that, we used the genetic variability represented by 19 genotypes with contrasting root architecture under full watering condition. And based on a previous project called Genopy, and this work was done by Virginie Bourrion from our department. And these genotypes were subjected to 2 weeks drought stress at flowering initiation. And what we found was some genotypes were not affected by water deficit. Here there are points in red. Here the value corresponds to the biomass loss when compared to the well water control. So here it means that it's not significant. The loss of biomass is not significant, and below this number, the genotype was affected by the water stress. So we tried to find correlation, traits that could explain why some genotypes were more or less susceptible to water deficit. And we found that in a counterintuitively mode, the number of secondary roots on the root system were negatively correlated to the resistance to water stress. And this could be due maybe to the management of competition for carbon with the nodules. Then we feel that it's now important to better identify the combination of root traits that could promote resilience, because they are, this data was done on only some genotypes, so we need to explore more genetic variability. And what we would like now to explore is the capacity of fruit for prospection in less dry areas. So here we have different shape of root systems. So then we would like to see if we can find an effect of the initiation or the location of the nodules in the root system. So here, for example, you can see that here the growth of nodules is favored, and here it's the development of nodules that is favored. We have more nodules. Whereas here we have the initiation of nodules in different wave that is favored, and here it's only one wave of nodulation that can be observed. It also concerns the compromise between the development, the growth of fruits and nodules that can allow hydromineral uptake at a lower cost in carbon. So here we can see that on this root system we have roots and nodules, but here we have more roots than nodules, meaning that maybe we have less nitrogen uptake, but we can get more water, so we have to find the good trade-off to allow a better plant growth. And finally, we would like to explore how synergy and antagonisms are possible, and especially with functional traits. So we don't want only to look for morphological traits on the root system, but also how the root system is able to uptake water, micro and macronutrients, and for sure the nitrogen fixing activity. So this work is ongoing in a project called Agroecology. And in order to better characterize this root system, we are quite fortunate in Dijon because we have unique devices that are called rhizotubes, which allow non-destructive access to the root system at high throughput. So this root phenotyping platform is called 4PMI, and you can find out more about it at the end of this link here. So here you can see, for example, the evolution of pea root growth, and the estimation of root biomass and nodule biomass of this plant. So you can imagine that we can easily find some growth traits of the root system and try to make the link between morphological traits and functional traits. So having confirmed that the resilience is genetically variable in response to water deficits, we wanted to identify the molecular processes that are involved in this response. And to do that, we set up an experiment under controlled conditions with a design which is quite similar to the previous one, meaning a 2 weeks water stress period applied at flower initiation, followed by a recovery period, a rewatering of only 7 days, because we wanted to capture the early responses. And in the previous result, I'll show you what happened at 15 days after. So we are just a little bit before. And we characterize the ecophysiological variables, as previously mentioned, and we carry out also transcriptomics and metabolomics of the root system. And at the transcriptomic level, at the end of the stress period, here, we had 400 differentially expressed genes in roots that were mainly involved in protein degradation, in development, in stress response. And after watering, you can see that only 20 genes remained differentially expressed with opposite expression patterns, meaning that the genes that overexpressed at the end of the stress were expressed when rewatered, and the ones that were underexpressed at the end of the stress were overexpressed after rewatering. And at the metabolic level, we can see that the metabolic composition of the root at the end of the stress was very different. Here you have the well water, and here the water deficit. And after 7 days after rewatering, the well water and water deficit samples are overlapping. So it means that at the molecular and ecophysiological analysis converge to indicate a total recovery of the root function after rewatering, although their structure is still very different. Then concerning the nodules here, the things are very different, because at the end of the stress, we also, more or less, we have the same number of genes that are differentially expressed between the control and the water deficit, 400 genes. But what happens after rewatering is that we have a great increase of the genes that are differentially expressed, with more than 3,000 genes. And these genes are involved in the same processes as mentioned before, but also in signaling, in secondary metabolism, indicating that the tissue are still in full regulation. And at the metabolomic level in nodules, we see that the metabolic composition of nodules remains very different from that of well water plant. So it's here we have the well water, and here the water deficit. And after rewatering, the samples are not overlapping at all. They are still different, although we can see that it goes in the sense of an increase of the glutamate and aspartate that suggests maybe a gradual recovering of denitrogen fixation. So here we can see that the roots and the nodules appear to have different response dynamics during rewatering. Early on, roots set up transcriptomic responses, enabling them to recover their uptake function, while nodules maintain the growth of their structure, but are not fully recovering their function. So this work suggests that it's necessary to characterize the recovery kinetics of the two organs, because we see that it's really different. We suggest that there may be a link between the recovery kinetics and the better resilience during a water deficit. So that's why we decided to characterize the dynamics of post-water deficit recovery. So we use this conceptual scheme where the value of a trait, so here, for example, we can imagine that it's plant biomass, and the value of the traits evolves over time. So when compared to the well water control. So for example, here we have a decrease during the water deficit. And then after rewatering, this trait will gradually recover. So recovery can be characterized by a latency time before recovery, an initial speed of rate of recovery, and a time to return to a stable stage. And delta here is the difference between the plant that have always been well watered and the plant that had a water deficit. So we work on this dynamic in two different genotypes. One was considered as being resilient because at the end of physiological maturity, there was no effect of the water deficit on the yield, while another genotype, Puget, was considered to be less resilient because it was not able to recover fully, and there was a huge effect on the yield components at physiological maturity. But at the end of the water stress, these two genotypes were affected similarly. So it means that they were different only for their ability to recover during the rewatering phase. So these genotypes were characterized at the ecophysiological, metabolomic, and transcriptomic levels via RNA-seq. And in the two genotypes, we observed very different recovery dynamics linked to our three parameters: the latency, here as the rate of the recovery, and also the delta. So we can see that in the more resilient genotype, in orange, Cayenne, the latency for the establishment of new nodules is associated with a less rate of recovery. And for the least resilient genotype, the Puget in green, we have a good nodulation reactivity that is combined with a too high rate of recovery that leads to an overcompensation, and that results in an additional carbon cost for the plant. And the carbon that is allocated to the nodules is no more available for seeds and for yield. So when we have looked at the molecular level, we have seen that we made some clustering, and it was combined with gene ontology analysis. And we were unable to monitor the kinetics of the molecular response in root and nodules of the two genotypes. And we've seen that we had a significant difference in the latency time between the two genotypes, similarly as what we've seen at the ecophysiological level. If you can see here, very quickly after rewatering, we observed a reduction in nodule energy metabolism and a recovery of growth mechanism after 3 days. Whereas in the less resilient genotypes, it happened only 6 days after rewatering. And concerning the genes that are responsible for responses to water deficits, they were still highly overexpressed in the first few days after rewatering, and growth mechanism did not recover until 6 days later. So as a conclusion, we can say that a best post-drought recovery is explained by a short latency period before activity starts again, and by the ability to adjust nodulation to the plant's nitrogen requirements. So now we can say that this work still requires an effort to integrate all the heterogeneous data in order to identify candidate genes for resilience with a better recovery capacity, and to find the molecular markers that could help for breeding. In addition, here we only worked on the plant side. We only focus on the plant component, but you know that concerning fixation, it's a question of interaction between the plant and the rhizobia. So we can also make the hypothesis that the rhizobia strain can impact the reactivity of nodulation after drought. So it's a new direction that we would like to explore. So working on the interaction between the genotype and the rhizobial strain to find the most reactive combination. Then another way of approaching the question of resilience was to look at how plants could react to multiple stresses, because in the context of climate change, plants are subject to recurring climatic stresses. And this is especially the case with water stress, because in some years, drought can occur several times during the same growth cycle. So this is the reason why we are interested in the memory of water stress in pea. And so what do we mean by memory? It's an acclimation strategy based on pre-exposing the plant to a first stress. And here Yeah, as you can see on this diagram, the pre-exposure to a stress induces an enhancement of plant defense response that can be more effective or faster, and thus a better tolerance to a second stress when compared to what we call a naive plant, which has never encountered any stress. So we first wanted to find out whether there was a genetic variability in the plant's memory of water stress in P. So to do that, we carried out studies under controlled conditions and also in the field with different water treatments. So we applied a single stress either at the beginning of the growth cycle or during the reproducing phase. We also applied recurrent stresses. So a first stress during the vegetative period and the second one during the reproductive stage. And we also applied a continuous stress from the beginning until the end of the growth cycle. We explored the P relative growth rate of the 5 different genotypes. These genotypes are spring P genotypes. And here you have the different types of stresses, either single stress, combined stress, or continuous stress. And on this graph, you can see the growth rate of the genotype as a function of the type of stress. We can see that some genotypes are more tolerant to water stress when the water stress comes early. So here it's the case of this genotype meeting that is growing better when water deficit occurs early. But when the water stress occurs later during the growth cycle, we can see that genotype astronauts react better, has a better productivity. And here, when we have combined stress, we can see that genotype saffron is able to have a better productivity, meaning that maybe is able to mobilize its stress memory better than the other genotypes. So then we wanted to identify the traits that are involved in each type of drought stress response by using an ecophysiological, functional, structural framework. So here it is. I'm not going to detail everything, but the idea is that the plant is divided into several compartments. So here we have the plant we have the aerial parts, the root compartment with the roots and the nodules. And for this compartment, we have detailed some traits. So for example, for the nodules, we have the number of nodules on the taproot or on the lateral roots, and we have separated the length of the taproot, the number of the lateral roots, etc., etc. And then by tracking the value of each of these traits over time that correspond to the vertical line, and as a function of stress, it depends on the column here. We found that some traits were more affected by the stress than others. And for example, in each box, when you see that it's colored red, it means that it's highly increased by the stress. And when it's blue, it's highly decreased by the stress. So by comparing at each period of watering and stress and so on, we are able to see that some traits are very involved in acclimation to continuous stress. It's a case of the taproot length and the number of lateral roots, while the number of nodules is mainly involved in recurrent stresses. So here it's very fast to understand something on this diagram, so don't hesitate to see the paper from Jacques from last year, where you will find all the information needed to understand everything. And so in a second step, we decided to work and look at the memory genes. The genes are supposed to underline the memory response of P. And what is a memory gene? It's a gene whose level of expression differs during the second stress compared to its level of expression during the first stress. And during the second stress, its level of expression must also be different from its level of expression during a single water stress at the same period. For example, here in this graph on the left, we can see that in blue, we have a memory gene that is underexpressed during a first stress period in orange. And then after the stress, the level of expression decreased to a basal level here during this rewatering period. And during a second stress, we have an increase that is higher than the upregulation during the first stress. And then when we have a rewatering, then the level of expression comes back to basal level. So we have different types of memory genes. Here we have an increase in increase, so we call these genes plus plus. We can also have a decrease during the first stress and a decrease even more higher, lower during the second stress. So these genes are called minus minus. And we can also have different type of expression. Some can be upregulated during the first stress and then downregulated during the second stress, and the opposite. And here we can also have some genes that were not affected at all by the first stress, but only were up or down regulated during the second stress. By opposition, you can see here genes that are not memory genes because they are differentially expressed exactly in the same way during the first and the second stress. So in our experiments, we found around 2,400 genes that were considered as memory genes in roots. So it means having this pattern, this one or this one. And this corresponds to 5% of the gene in the roots and 9% in the nodules. We have to note that this proportion is consistent with different studies in maize or in Arabidopsis leaves. And when we look at what type of gene we can find, most of the memory genes in roots are related to this pattern here, meaning that most of them are not really differentially expressed during the first stress and are especially expressed during the second one. And we also have this proportion, that meaning that we have this one, an opposition of the level of the expression, that genes that were upregulated during the first stress are downregulated during the second one. While in nodules, most of them, than 90% of the memory genes are really related to this kind of expression pattern. So it's the main differences between the two different organs. So now under process is the identification of candidate genes for P drought stress memory. So we want to identify the major regulators using a gene co-expression network approach. And then we want to correlate with ecophysiological traits. And we hope we will be able to identify key genes involved in root stress memory. Okay, so now it's just a quick last part of my talk, and it's to show you the importance of soil microorganisms in legume resilience to water deficit. So the question we ask ourselves were whether there was any point in looking at microbial communities as a whole in a species, in this case it's P, that establishes symbiosis with microorganisms to facilitate its water and mineral uptake. And so we wanted to assess whether microbial communities could play a role in resilience. And to do that, we set up an experiment to dilute the diversity of microbial communities in the soil And this resulted in having 3 levels of microbial community diversity: a high diversity, a medium, and a low one. And 2 genotypes were studied: a wild genotype establishing symbiosis with rhizobia and mycorrhizal fungi, and the mutant that was not able to make a symbiosis neither with the mycorrhizal fungi, neither with rhizobia. So we applied a water deficit at flower initiation during 2 weeks, and after the 2 weeks, some plants were harvested and others were rewatered until physiological maturity. And what we observed is that there was no effect of the level of the microbial diversity on the resistance to water deficit. And it was the case, whatever the genotype, the wild type or the mutant. So here you can see that you have the 3 levels of microbial diversity: high, medium, low. Under well water condition, here it's underwater deficit, so we can't see anything. And what we observed was that during the rewatering period, we see an effect of the level of microbial diversity, because at physiological maturity, we can see that for the plants that were grown under a high level of microbial diversity, plants had a higher biomass than the ones that were grown under a low level of microbial diversity. And this was also the case when we looked at the seed biomass, so at the yield component. It was higher under water deficit with a high level of microbial diversity than under low microbial diversity. And we found exactly the same pattern with the wild type and the mutant not carrying symbiosis. So this work has shown that higher diversity of microbial communities confers a better recovery of P after drought. So it means that a minority microbial species can have a really high added value in agro-ecosystem. It's not only a question of symbiotic communities. So it means that we also need to look at the microbial community as a whole and not only look at the symbiosis. So just as a quick conclusion, I wanted to remind you a few key points from my presentation. First, the nodulated root system plays a major role in peas resilience to water deficit. Then finding the right cost-benefit ratio between root and nodule production is essential under fluctuating water conditions. Then to find resilience traits, we need to take into account the dynamics of response to stress, whether single or repeated, recurrent, and the ability to recover from stress. And finally, the role of microorganisms needs to be considered in a more comprehensive way, whether symbiotic, rhizobia or mycorrhizal fungi, or also non-symbiotic. So I would like to thank you for your attention and thank my colleagues from the Agroecology team here in Christmas mode. People from the 4PMI high throughput phenotyping platform, and also bioinformaticians, geneticists, physiologists, microbial ecologists, microbiologists from Dijon who participated to this research, and also colleagues from other labs, in particular for transcriptomics and metabolomics approaches. So now don't hesitate to ask me any question. I will be happy to answer them.
Thank you, Marion. It was for the very interesting presentation. I will start with one question that arrived via chat for somebody that didn't want to actually talk but preferred to write. And the first question is there a correlation between normal leaf and the Aphylla leaf types to the ecophysiological response?
Yes, actually, when we compared two different genotypes, one was Aphylla and the other one was classical one, and we observed that the quantity of water that was lost by evapotranspiration was more or less the same. Maybe because the dendrites are also transpiring a lot. And this was observed. We quantified the number of stomata on the tendrils and on leaflets, and actually we have a lot of stomata on the tendrils also. So we were not able to see any big difference.
Okay, interesting. Other questions? Let's see if somebody raises their hand. Well, while our crowd think a little bit, could you tell us a little bit more about the phenotyping system, the rhizotubes? From the images, it looked like that the roots were growing in some kind of gel. Is it possible?
Not really. Actually, it's a special device. The root system is growing between a plexiglass and a membrane. And this membrane, behind the membrane, we have a substrate. It can be inert substrate if we want to be sure to control the nutrients and the water and so on. But we can also fill it with soil. And this membrane lets the water, the nutrients, and the microorganism go through. So it enables us to see the root system without any soil particle. And as you have seen, the picture is very clean, so it's easy to get the root traits.
Okay, thanks. And a question in the chat. Do you know the time frame in which the plant switches from inorganic nitrogen uptake to N2 fixation?
Yes, actually it depends on the quantity of nitrogen in the seed and on the growth rate of the seedling. So, for example, in the varieties that we are using that are cultivated, usually fixation arrives after 2 weeks. So it means that all the storage, the nitrogen storage of the seed is over. And so a first wave of nodulation happens. Initiation of nodulation happens after 10 days, but we can say that the fixation is functioning properly after 2 weeks usually. I don't know for the wild type, but at least for the cultivated accessions, it takes this time. After the more the seeds are big, the higher we have nitrogen storage, and so the less how to say, the more we have nitrogen storage, the later the initiation will occur. The initiation of nodules will occur. Yeah.
Okay, there's a follow-up question from Nicola, which is the author of the previous question. Do you also determine N2 fixation using 15N labeling? I have no idea what
Yeah, it depends actually. We use stable isotope labeling when we have a mixed type of nutrition, because we want to differentiate the nitrogen coming from the soil or from the nutrient solution from the one coming from the denitrogen fixation. But in the case we are only having denitrogen fixation, we don't need isotope labeling, and we are just comparing the quantity of nitrogen at the time when the nodules are fixing, and at the time we want to measure it. So.
Okay.
I don't know if it was, yeah, okay.
Yeah, I'm happy with the answer. Other questions from the participants? Okay, so it appears that, no, wait, there's somebody raising their hand. Paolo.
Hi, Paolo.
Hi, Marion. Hi. Thank you. I found your webinar quite interesting. I would like to know, could you tell us a little bit more about what is microbial diversity in your study? I mean, is it just defined in terms of amounts of general microorganisms, or is there a way just to focus more on putative, useful microorganisms in such a study, or it is largely unknown and one can't do that?
Yeah, actually, what we did for this experiment, we extracted microbial communities from soil. So it means that we had the soil, we put water in, and then from this solution, we extracted the microbial, what we call the microbial communities. Okay, and then these microbial communities, we had two solutions. We keep like that, and then we inoculate into sterile soil, or we diluted it so many times so that when we do that, we are losing some microbial species. So we only keep the major species in the soil, and we are losing the species that are in minority. And we then, once we did this dilution, we re-inoculated, and then we grow pea in this soil, etc., etc. So it's not exactly real soil because we re-inoculated, and it's a little bit different, but still, it's not only a synthetic microbial community with only 10 or some species. We had thousands of species of microbial species in this experiment. So it can be more related to what can happen in the field than when we only use some species that we inoculate by 3, by 5, or sometimes by 10. But
did you proceed to any kind of quantitative measure, like after metabarcoding and measuring Shannon diversity, that kind of things?
Exactly, yes. We measured that to be sure that at least our experimental diversity, our diversity experiment was okay. So we just checked that at the beginning of the experiment to be sure that we had an effect. And then we also characterized the level of diversity throughout the experiment at the different levels. It's not published yet, but yes, we characterize the level of diversity and who are the species that are maintained or that disappeared during the experiment.
Okay. Questions or follow-up on this topic? Okay, so it appears that we reached the end of this webinar. I thank again Marion. You were very, very interesting, and you gave us a lot of useful information. This webinar goes on our YouTube channel and on our website. So take a look at those. Keep those under your very attentive scrutiny because a lot of news is passing by these days. Okay, thanks,
Okay.
and see you in a month. Thank you again, Marion.
Thank you. Bye-bye.